All right, terrific. So, um, hope my slides are showing. So, I have a very exciting company to tell you about at a very exciting point in its life. So, Angiex was founded by leaders in the field of angiogenesis. That's the formation of new blood vessels, with the aim of creating a universal therapy for all solid cancers that would uh, kill tumors by eliminating their blood supply. And they discovered a way to do this about 23 years ago, and we've been developing it ever since. The biology turns out to be perfect for antibody-drug conjugate therapy, and it enables us to attack two compartments of the tumor, not only the tumor blood supplies, but also the tumor cells, because the same biology that enables new blood vessels to grow enables tumor cells to invade and metastasize. So we made our drug. We brought it into the clinic a year and a half ago. We've completed our initial dose escalation quite recently with outstanding results. So the drug has been very well tolerated. We've got a very broad therapeutic window. It looks like approximately a threefold range between where the drug begins to be effective and where we begin to see severe adverse events. And we expect a very large value inflection in the near future. And there's an opportunity to enter at a very good valuation. So this slide illustrates the vascular mechanism of action. So what our drug does, it eliminates all the small vessels within the tumor, but it also eliminates the large vessels that feed and drain the tumor. That shuts off the supply of nutrients and oxygen to the tumor. It leads to a mass necrosis in the middle of the tumor. And cells tend to survive on the periphery of the tumor, but with a significant wounding, which leads to scarring, as well as large death of tumor cells here. And in addition, TM4SF1, because it enables invasion and metastasis, it essentially enables what's called the epithelial to mesenchymal transition in tumor cells. And in late stage cancer patients, most cancer cells have undergone this transition, and so they're high expressors of TM4SF1. And so that's why TM4SF1 is very highly and broadly expressed across all solid cancers. It basically is what enables cancers to become lethal. And so we have the opportunity to kill off the most malignant tumor cells uh, and potentially kill off all cells capable of invasion and metastasis, which are thought to account for 90% of cancer deaths. So, great opportunity to potentially dramatically reduce cancer mortality. So this is what we've done through our dose escalation. So the lowest five dose levels all cleared safely. The top dose level, we dosed three patients. Two of them had only grade one, which is mild adverse events. One of them had a grade three, which is severe adverse event. And so to understand the toxicity, we would have had to add more patients. But by that point, we had learned that we needed to alter some other characteristics of the trial. And so we've gone back to redo these. And the most important thing we learned was that CT scans were not giving us the efficacy readout we needed. And so we added FDG PET imaging, and now we're essentially redoing the top three dose levels with PET imaging and also with some adjustments that we think may eliminate severe adverse events. And so this illustrates our efficacy. So by CT scans, we've essentially eliminated tumor growth. So this distribution, it's really essentially a random distribution around zero, centered on zero, and approximately equal numbers of tumors on each side. But what we believe is happening is that the vascular mechanism, when we kill all the tumor blood vessels, that induces a coagulative necrosis and a lot of fibrin deposition, which ends up healing by scarring. And so we convert the tumor to a scar that's the same size as the original tumor. And as a result, on CT scans, you always see a lesion that's the same size as the tumor was at the time of first dosing. And if our drug was ineffective, then the bulk of these dots would be in this yellow range. So we're quite confident that our drug is doing something beneficial for the patient. And then this slide illustrates our therapeutic window. So four patients have experienced severe adverse events. Those are the four to the right of this blue line. So basically the four at the highest doses. So the two patients, these are both diabetic and obese patients who got by ideal body weight, got the two highest doses in the study. And then up here are two hypertensive patients. One had a systolic blood pressure of 158, one of 161. And it appears that with hypertension, the severe adverse events can come in at a somewhat lower dose. But if we put a maximum tolerated dose here, maximum dose here in the hypertensive patients and here in the non-hypertensive, we hope we can eliminate severe adverse events. And this gives us a very broad range in which we think we're effective. And so we believe we've got a window where we can be effective against every tumor of every solid cancer. And just for background, so each of these is one patient, and the average patient has about four or so target lesions that are measured, plus another four or so non-target lesions. These are all metastatic advanced cancer patients who have failed all approved therapies. So the most difficult patient population, and we really haven't found a tumor that we don't impact in a favorable way. So we've got an outstanding team. Uh, I myself, four-time entrepreneur. I was a scientist at MIT, Berkeley, and Harvard. Wrote a best-selling book, successful in several areas of life. I'm aided on the executive side by Ian Dukes. He led business development at Amgen, led business development at Merck, and has been CEO or chairman of a number of companies in which OrbiMed was an investor. Our chief medical officer led oncology research at one of the Harvard hospitals before moving to industry and serving as a multi-time CMO. Our manufacturing and quality leaders, who are very important, are the best in the business for ADCs. They led manufacturing quality at the top two ADC companies for many years. Between them, seven approved ADCs, $70 billion of corporate acquisitions. Our chief financial officer, longtime investment banker, founder of the healthcare practice at Goldman Sachs, and then five-time CFO. And our scientific founders made the top two discoveries in the field of angiogenesis. In conclusion, we have what we think is a terrific investment opportunity. We have an open convertible note, 20% discount, 10% interest rate, $100 million cap. For background, there's been $90 million into the company over its life. So with the note, the cap ensures that you're essentially getting the company at cost. So it's like a seed level evaluation in a clinical stage company with a working drug that has potential to serve a vast market. And the stock is qualified small business stock. So if you hold it long enough, it can be capital gains tax free. And we're on the verge of a value inflection. So we think the PET imaging will demonstrate that we're killing most of the tumor cells in the tumor, and it will enable us to find the optimal dose. And once we prove that we're killing off tumor cells, we'll be able to keep the patients on trial and demonstrate a survival benefit. And that will lead us toward approval pretty rapidly. So in conclusion, this is a picture of my mother. She was diagnosed with cancer at age 22 when she was pregnant with me. She died at age 33 when I was 10. When I was young, whenever I complained something was unfair, she would respond very sternly, "Life is unfair." And I didn't understand why she was so stern on that point until after she had died, and I realized how unfair life had been to her. Now I'm meeting lots of cancer patients to whom life has been unfair, but we have a way to remedy that. And right now, we have what looks like a very clear path to approval, saving many lives. And the only thing that's limiting us is capital in. So investments can accelerate our path to approval and save more lives. So I hope you'll join us, uh, and I think we'll deliver terrific investment returns, but also have a huge impact. Thank you.
Great, Paul. Great presentation. Isaac, did you have any questions there for him?
Um, Sorry if I missed it, but just could you go back a little bit further into kind of use of funds and what um, the raise?
Yeah, yeah. Um, so, so we've been following more or less just-in-time investment strategy because we've expected a major value inflection would be imminent. So we've kept the valuation low to make, to ensure that we wouldn't fail to fundraise, but we've kept the round sizes small to minimize dilution. And so we're basically continuing to follow that strategy. So we're just looking to get, many investors are waiting for proof of efficacy, which is primarily measured as responses. So they want you to prove that you're killing most of the tumor cells. And most drugs can do that with CT scans. And we started out with a fairly standard trial design that would rely on CT scans to demonstrate tumor cell killing. But in fact, what we found is that the vascular injury appears to lead to scarring, which is actually a great outcome for the patients because if it just eliminated the tumors, you could create perforations or things that could cause health issues. And healing by scarring is a very kind of conservative outcome that ensures nothing bad happens. It stabilizes the tissue. So it's good for patients, but it's makes it harder for us to demonstrate that we've killed tumor cells. But the PET imaging should achieve that for us. So essentially we've got a ton of investors waiting to see the PET imaging data, and we'll get those scans in April, May, and June, and we're just looking to maintain runway as we gather that data. So we need to bring in $1 million a month approximately to, not have our runway run down. But we can raise more than that.
Thank you. That helps.
Great. Paul, can you talk about the next steps here with the clinical trials? What do you have to do to achieve phase one?
Yeah, so this is really, the next few months will be completing our phase one. So basically, if I go back here, we're redoing these dose levels with PET imaging, and the other changes we've settled on the Q6 week interval as the best. And we've also shifted from actual body weight scaling to ideal body weight scaling. So that's what uh, shifted. We found these obese patients, so this is by ideal body weight scaling. So with ideal body weight scaling, the obese patients move out to the right. So one of these was, I think this one was a 310-pound woman. And so with actual body weight, she would get 3 times the dose of a 103-pound woman. And uh, that's not necessarily that's not for us the right way to scale. So we found that our obese patients would be the ones who would get these adverse events. So we've shifted to ideal body weight, and so that along with the Q6 week dosing should eliminate the severe adverse events as well as these maximum doses. And then the PET imaging should demonstrate that the tumor cell population has declined precipitously. And with that data, So once we find the right dosing regimen that gets us efficacy with safety in all patients, that determines what's called the recommended phase 2 dose. And, with that, we can proceed to phase 2. And so we have lead indications identified. Our first indication to approval is angiosarcoma. Our second is pancreatic. Those are high unmet need cancers where we can get approval in a phase 2 trial. And in angiosarcoma, because there's no approved drugs for it, we don't even need a control arm. And there are orphan indications with very rapid paths. So we can essentially start a registrational trial in 2027 and potentially finish it in 6 months. So we've got a very rapid path to approval. If we bring in money quickly enough to get the manufacturing and quality work done, we can get to approval in 2028. And we can help an awful lot of patients. So capital in is really, and the ability to start manufacturing and quality work is really drives the timeline to approval and lives saved.
Great. And what do you see as the exit? Is it upon FDA approval or before or after that?
Well, so we're expecting to do an IPO in 2027. At least we're preparing for that. We'll be PCAOB audited this summer. And we think with the good clinical data and with FDA validation through an end of phase 1 meeting later this year, that'll chart out our path to approval and designations like fast track, orphan, breakthrough designation, breakthrough therapy. We'll be in a good position as far as clinical data to support an IPO process in 2027. Um, uh, so we're not looking to flip the company quickly because we have potential for a huge market and we want to get to approval and let the market see commercial uptake. And we also, I think, as I mentioned, we're qualified small business stock, and we'd like to give our shareholders an opportunity to hold the stock long enough to enjoy that tax benefit.
Okay, great. And that's our 20 minutes. Thank you so much.
Thank you.
Let's have the Solaris next. Randy, if you'd like to launch your slides and kick off and ready.
Yeah, absolutely, Hall. Thank you. All right, here we go. Let me pull up my slides. And
Looks good.
okay. Hey, hello everyone. I'm Randy Hubble. I'm CEO for Solaris Endovascular. We've got a $5 million bridge round that will lead to one equity round that will get us to exit. You're going to hear me talk about exit quite a bit because this company is specifically set up to be acquired by a large multinational. We do have a due diligence report completed by Keiretsu Forum Northwest. We've had other significant number of other angels invest in us. What's going to be different about this presentation and maybe some of the others that are earlier stage companies is we have a recurring revenue. Solaris SX, our base technology, is going to do about $5 million of sales outside the United States this year. Our next generation product, Solaris DE, we have clinical data, phase 2 data that's spectacular. It's going to create a new category, a new endovascular category. And this is a shiny object. This is driving significant interest from strategics. I've got a strategic that's very involved and engaged right now, waiting to hear back from a meeting with Gore, BD, Cordis, Abbott all next week at the LSI meeting. I've had multiple exits. We've been very capital efficient. I set the company up in such a way where I formed a Delaware C Corp. I used equity to acquire the assets and to move fast through some significant milestones in the first half of this year. Every month we got something big going on. We've lowered the valuation cap from 50 million down to now it's at 35 million this month and 40 million next month. So great opportunity with our equity round being in the 60 to $70 million range. That's could be a great discount. So what do? We have technology for the endovascular space, the peripheral space. For dialysis access patients, they have their artery and vein tied together, and it's accessed with a needle, and the blood's taken out and put through a kidney machine, and then it's put back. That happens, that flow rate is very high. The shear force, the pressure, the turbulence, particularly on the venous side, creates neo-intimal hypoplasia. Think of it like scar tissue. We also call it a lesion or a stenosis. All of it's the same thing. What it is, it's a narrowing of the vessel. That's what we fix. Peripheral artery disease, the same disease that folks have in their coronary artery, atherosclerotic disease. It happens all over your body, and then when it's in the lower legs, that becomes highly problematic. So our Solaris SX is our first product. It is currently treating both of those areas, dialysis and peripheral artery disease. We've sold over 15,000 units to date. We've got approval in 45 countries, including MDR CE mark in Europe. The second program is a combination product. It takes the Solaris SX and it puts sirolimus, a terrific anti-proliferative safe drug, on the edges of the stent. That becomes a combination product, and that's in phase 2, and we're on a path to approval in the US. We're focusing on the US with Solaris DE. We're not going to bring SX to the US because the DE data is just so unbelievably incredible, better than regular covered stents. We are taking share, though, where we compete overseas from our competitors, which are going to be Gore, BD, and Merit, because we have a unique combination of great radial strength plus flexibility. This is a nitinol self-expanding stent. It's covered with electrospun PTFE. Only us and Merit have the electrospun PTFE. Gore and Bard have the expanded PTFE, which is a couple of decades old. We have data. We've done this in the iliac, the artery in your groin. At 12 months, we have 95% patency. Patency meaning the vessel is open. Very good data. With the dialysis access patients, the standard of care for years has been balloon angioplasty. So what that means is a patient shows up with a flow rate that is diminished. It's because of a lesion. They go to repair that lesion with balloon angioplasty, and at 6 months, half of those patients are coming back. Half of them are coming back because that repair has failed. It was not durable. With our covered stent, and it's also very similar for the other 3 competitors, Gore, Bard, and Merit, we are able to improve that patency rate to about 75%. So about a 25% failure. So at 6 months after you repair it with a covered stent, you have patients that 25% of them come back where that repair did not stick. It was not durable. And that doesn't happen in the middle of the stent. It happens on the edges. And that's what's called edge restenosis. So when you put a covered stent in a dialysis patient to repair a lesion, 1/4 of the time, 1/4 of the time, it will narrow and fail on the edges. So what you're looking at here is a nitinol metal stent. The contrast is the dark piece in the middle. On the white milky parts on the edge of the stent, that is contrast that's not flowing or not flowing very well. And that's a stenosis. That's low flow rates. That's a problem for dialysis patients. We fixed that problem. Here's how we did it. We took that base stent and we took the metal edges and have a drug-eluting on there called sirolimus. Sirolimus is the same drug that was used with the first ever drug-eluting stent called Cypher for the coronary. So back in the day, bare metal stents were put in. Patients came at 6 months, about 25 to 30% of those patients came back with a restenosis, an overproliferation of the vessel and a narrowing that caused a problem. When we put a drug on it, we brought that down to 3%. I was the launch leader at J&J for that technology. We hit $1 billion of sales in 8 months with Cypher. We're looking to have the same impact here on the peripheral side with this Solaris DE product. And we have data. We have finished enrolling in a 115-patient phase 2 study. We have the first 60 patients have hit the primary endpoint of 6 months. These are dialysis access patients. We're measuring target lesion primary patency. And as a reminder, with balloon angioplasty, you're going to have about half of those repairs that were not successful at 6 months or failed. With a covered stent without a drug, about 25% failed, or 70 to 75% patency. We had a 95% patency with the first 40 patients that have received this drug. So 40 patients received it, 20 received balloon angioplasty. And the 2 that failed were because of geographic miss, meaning the doctors didn't put the drug on the lesion. They went past the lesion into the central vein, and it washed it downstream to the lungs. So we are very optimistic about this. We'll have the full data readout in June of all 115 patients. The picture you're looking at on the right will look just like the Cypher pictures at 6 months, 12 months, 18 months, very durable. What you're seeing there is that black lumen is an open vessel. The stent is up against the wall. You're not seeing any scar tissue. This is fabulous. We are expecting to see the same type of wonderful pictures at 12 months as well. So the data drives the day, and this is what's got the strategics interested. Our market is a $1.5 billion market today. We're going to grow that and double it by 2030 with the introduction of our covered stent, because we'll charge at least twice as much. The covered stents today are well reimbursed by Medicare. Medicare covers all dialysis patients' cost. Their ASP for a covered stent without a drug is $3,000. Our price for this will be about $6,000. This company was set up for an exit. That's all I do now is exit. So I'm not interested in an IPO. I'm not interested in running a company. I looked at this market and I saw that these large multinationals are heavy into the endovascular space. They're spending millions of dollars on infrastructure, but they have no covered stent. Only Gore, Bard, and Merit have them, and they're not good acquisition targets for these companies. So this is a tuck-in. This is a pure play. This will be acquired. We've got 2 companies under NDA. We've got others that we've had first contact. We'll have second contact with them. We are in due diligence with 2 of them today. One of them, we're expecting that we'll have some progress here this month. We are moving very fast. We are hitting a lot of milestones. This bridge round of $5 million takes us through 2026. What we're doing is we have submitted for breakthrough designation. We expect to hear back at mid to late April. We'll submit part 1 of our IDE submission, the two-part submission. That will go in at the mid-April, into March. We've got FTEs, the strategic that's interested in us, asked us to stand up the company, because I started very fractional and virtual to save money. And so now we're in the process of bringing on those FTEs. And then you'll get the full readout in June of our phase 2 study. Quite exciting. And then with breakthrough designation, we will have multiple meetings, either official pre-sub meetings or sprint meetings or informal meetings. And then we only need, after this convertible round, we'll do an equity round at the end of this year or beginning of next year, $17 million. This is a very straightforward play. IDE approval, 1 pivotal study, 1 PMA. Once we get the data on the pivotal study, the acquisition window opens up, and we should be acquired for about $550 million. This is the launch that supports this. So I do expect that I will not be commercializing this. The conversations I'm having with the strategics is that they would acquire it before commercialization so that they can use their big engine to commercialize this. But the revenue forecast does support that $550 million, as does the NPV, net present value analysis, all supports that. And also comps. We have multiple comps that support it. So I've done this before. I had 5 years at Boston Scientific, 13 years at J&J. Cardiva was my first company after 13 years at J&J, and that was acquired by Haemonetics for $510 million. Carmel was I went from Cardiva to Carmel. That was a spin out of Carnegie Mellon. 7.5-year journey and got it public on the NASDAQ. in the summer of 2023. Dr. Marco Costa is an interventional cardiologist. He is a scientist with a PhD. He's also got an MBA from MIT, and he's run hospital systems for United Healthcare. And he's also the CEO for Cytech Medical, the company that we acquired this technology from. So in conclusion, we've got $2 million in the bank. We got another $1 million that will come in by Friday. We just did some document revisions. All of that will fall under most favored nation clauses. So those $2 million already in will automatically receive those benefits. 25% discount. We do have a valuation cap of 50 million, but we've lowered it to 35 million until the end of this month or the next $1.3 million. Then we'll have more milestones. We're going to, get this $1 million in. We've got an independent director that's coming on board that has multiple exits, just had the largest pre-commercial medical device exit in the history. He's going to be coming on board. So the valuation will go up to 40 million for April and 45 million. Then in June, it'll be 50 million. So we're looking for folks to help us fill out this round. Thank you.
Great presentation, Randy. Francisco, if you have questions, you're welcome to put them in the chat box. My first question is, why have not the large strategics like Boston Scientific, Medtronic, or Abbott entered this market yet? And why is now the right time for them to do so?
Yeah, so they have not had the technology. So Gore has had the technology for years. Bard developed it as well, and Bard was acquired by BD. And so they have not had the nitinol PTFE capabilities. And certainly, the combination product is unique to us. The other competitors don't have it. And, it's not uncommon, Hall, for these companies to enter a new market through an acquisition.
Right.
So
That makes
that's the ecosystem for medical devices. I'm just leaning into that ecosystem. They expect, startups, early stage companies to develop technologies that they can acquire and get into a new market.
No, that's great. Well, Solaris is an up and running company that has revenue from outside the U.S. And so what lessons did you learn from, that revenue stream outside the U.S. for strategy for physician adoption, reimbursement, and other aspects of it?
Yeah, so what we've learned from our OUS sales is, it's all through independent distributors. It's very fragmented. We don't support it very heavily with commercial support. And what we've learned is that the technology is very competitive with the other products and has features and benefits that are superior. We're number one market share in Brazil, number one market share in Turkey. So it's, you should look at it as a test market. And what it will allow us to do is when we strike a deal with a strategic, they don't have to wait until Solaris DE is approved in the U.S. They can start getting revenue outside the U.S. It's a validated technology. We are approved in 45 countries. We're only selling in 10. So the idea would be for whoever does a deal with us, they would distribute that product starting this year and use their vast, commercial infrastructure in all 45 countries to really drive the sales. But they're able to do their due diligence and see that it's a very competitive product to the ones that are out there today.
And my last question is, how much more money do you have to raise after this round to get to FDA approval?
So it's 17 million so it'll probably be like a $20 million equity round.
Okay. Oh, I see. Okay, great. That's it
we only have so far we've got only 4 million. So I took $1 million of seed money from the Cytech, the company I acquired this from, and then we've got 2 million in the bank, and plus the $1 million that's coming. That's only $4 million. So we're extremely capital efficient, and we'll be able to have with the milestones we're hitting, we should have a pre-money on that equity round of somewhere around 60 to 70 million.
Okay. No, that's great. It's a great set of numbers there for you. It's very efficient and very fast compared to most others. So that's great to see as well. Thanks so much. I think that's it for the questions right now. And let's move to our next presenter. We have with us next Novuson. Stu, if you want to launch your slides and kick off, I'm ready.
Sounds great. Thanks, Hall. Everybody see that okay?
Yes, we can.
Cool. Well, I'm Stu Mitchell. I'm the founder and CEO of Novuson, where we are clearing the way for safer surgeries by using sound to eliminate the excessive burning of patients' tissue and literally clearing the smoke screen, making the surgeries faster and more precise without risking the health and safety of the healthcare workers or the patients. So have you ever wondered what happens during a surgical procedure? Well, this is something that was brought to my attention by doing research at the University of Washington and working with surgeons using high-intensity focused ultrasound and current technologies preparing our animals for the studies we were doing. So current electrosurgical devices, whether radio frequency or ultrasonic, operate at extremely high temperatures. They cause excessive damage to the patient's tissue, and they put smoke in the operating room. These devices lead to about 250,000 complications per year. They increase operating time up to over 90 minutes per injury, and this results in long-term cost for the hospital systems. About $215 billion per year in the US alone. These devices that are currently on the market, two main ones are the Ligasure by Medtronic and the Harmonic Ace by Ethicon. They operate at well over 100 degrees Celsius, and they operate on a principle of heat conduction, which is the most inefficient way of putting energy into tissue. It's why they burn tissue. It's why they have excessive damage and cause smoke. Novuson's technology is direct therapeutic ultrasound. It's a more efficient way of putting energy into tissue. We operate at significantly lower temperatures. We have literally no thermal damage, less than a half a millimeter of thermal damage, and we do not make smoke. It's a more precise way of putting energy into the tissue. Here's Novuson's technology being used in an animal model. We use the ultrasound energy. We deliver it. It basically operates from the inside out. So we transmit the energy in, it meets in the middle, goes back out to the outside transducers. As you can see, we clearly weld the tissue together and we cut the tissue, all using ultrasound energy. Why the DTU wins is because our features are we have lower temperature, we have no smoke, and we have higher precision. This results in fewer complications, shorter operating times, and faster recovery. Compared to the other devices on the market, the Harmonic Ace and the Ligasure in general, we are significantly faster in sealing time, the degree of thermal damage that we cause, and we do not generate smoke, and we can operate we're fully functional in blood or other fluids. The other technologies do not do that. And we're building a platform technology here at Novuson. Right now, we're working on our laparoscopic general surgical instruments. We can make devices that can go into robotic surgical systems or endoscopic surgical systems, and of course, we can be used in trauma as well. The total addressable market for electrosurgical devices is $10.2 billion globally. We're looking at a specific segment of that, which is in women's health and ambulatory surgical centers, looking at doing a procedure called a salpingectomy. That's $110 million by year 3 for our segment. We have 2 revenue streams for our business model. One is the laparoscopic general surgery devices, which is a razor blade model. The handset is a single patient use disposable. The generator is capital equipment. These are priced along the midline of everything that is currently on the market. On the robotic and endoscopic surgery side, this starts out as an R&D collaboration where my engineering team designs a transducer technology for their surgical system. And once they've tested it and they like it, it becomes a licensing and royalty deal. The important thing on this slide is the unit economics. So for the laparoscopic, this is the razor blade model. We're looking at generating by 2030, we'll have 3 devices on the market, a 3 millimeter, a 5 millimeter, and a 10 millimeter in diameter. And on average, they will generate about $100,000 per surgical suite that we have these devices in. And that's in total, when we get all of our accounts in, we'll have about 1,000 surgical suites by 2030, working with 10 direct sales representatives. on the laparoscopic general surgical instrument side. We're initially going to focus on states where they have passed laws that require the mitigation of surgical smoke, and focusing in particular on ambulatory surgical centers that specialize in women's health. Our go-to-market traction to date is we've demonstrated our technology in numerous animal models, demonstrating that our thermal spread is significantly less than everything else on the market. We can seal and divide arteries up to the same diameter that everything else on the market can. We meet the FDA requirements of 90% efficacy and burst pressure of greater than 240 millimeters of mercury. We also have a letter of interest for $1 million investment from Salis, a $2 million letter of interest pending from BM. We've done a robotics collaboration with MedBot, and we're in discussions with EndoQuest about another collaboration, and Aesculap is looking at partnering with us. So a clear path to market for us. First thing that we need to do is get our regulatory studies. So there are no human study, or regulatory clearance, I should say. There are no human studies required for this. It's a direct 510(k) pathway. We've met with the FDA before the pandemic, and we're scheduling to meet with them again. Again, standalone surgical centers. We're going to work with early adopters, professional societies, and go out and direct sales. First indication is in women's health. Our beachhead procedure is a salpingectomy, which reduces the risk of cancer by over 50% in patients that are at risk. Next milestone is regulatory clearance. We just had our kickoff meeting with Medal Consulting, a medical device specific consulting company, for regulatory approval. We're targeting our pre-sub meeting, our second pre-sub meeting in May, and we should have 510(k) submitted by August of 2026. We're basically mitigating risk by making sure we're aligned with the FDA. We have a very good V&V plan in place. We've got risk management with a traceability matrix based on ISO 14971. Our DHF and QMS are just about completed, and we have a nice E-Star plan going forward. By 2030, we'd hit 281 in revenue. That's good. There are 3 devices on the market and doing 4 strategic collaborations in surgical robotics and endoscopic systems. Potential exits. These are companies that could potentially acquire Novuson or partner with us, and how it would increase valuation along the way. Currently, we're valued at $12 million. That's based on a 409A valuation. And you can see as we reach the different exits or different stages, like 510(k) clearance, getting our other devices on the market, valuation increases, or the exit value increases as we go. Trade comps, these are all the companies that recently that have been acquired by the main players in the market. Most recent is Hologic acquired Boulder Surgical for $160 million in 2021. Management team has experience in business, technology, finance. In particular, I'd like to point out Mart Pearson, who launched one of our main competitors' products, a product by Medtronic called the Aquamantis. He launched that, and he's very familiar about taking products into surgical suites. Marco and Mary have also launched medical products before, and Gene and I had a business that we grew from 10 people to 110 employees and sold it about 10 years ago. Board of directors are experienced business and financial experts. Medical advisors are surgeons in robotics and other surgical specialties that need a smaller, safer device. And in this next couple of weeks, we'll be bringing on 2 new medical advisors. One's a transplant surgeon from the University of Washington who's concerned about being able to remove donor organs without excessive tissue damage, and Michael Chody, who's the chief of surgery at Banner Health Systems. We've brought in about $7.3 million in equity to date into Novuson, $3.2 million non-dilutive grants is what we started the company on. We've met numerous milestones on the way. We currently have a Series B funding open, $3 million preferred equity that we're raising in 2 tranches. The first tranche, we have 563,000 in already. We're looking to close the rest of that out by the end of this month. And the second tranche will be led by Salis and BM, which would potentially take the entire round, if not oversubscribe it. All the fundings that we're bringing in, the total 3 million, will be used for regulatory clearance of our 3 millimeter device, initial market launch of the 3 millimeter, and final development of the 5 millimeter and 10 millimeter. And this is the breakdown for what those funds will be used for. We have numerous patents, 18 in total, US and international. We just received a new one last week. And this is a quick summary. 1 million preferred equity open. That's just about closed. 12 million pre-money. Letter of interest from Salis and BM. 563 invested so far. We've got 325 soft circled, so we should be able to close that out by the end of this month. And we brought in about 10 million in all in capital so far in Novuson. I've put in about 600,000 myself. And most of the funds bringing into this round are all for B&V and regulatory clearance. Happy to answer any questions.
Great presentation, Stuart. My first
Thanks.
question is you are targeting a $281 million revenue stream after FDA approval. After FDA approval, how much more capital do you have to raise to achieve that revenue goal?
We're looking at bringing another 10 to 15 million mid to late 2027.
Okay. No, that sounds good. And what valuation do you think might be on it at that point?
At that point, we should be valued around 50 million.
Okay. Okay. And so you're having to change the surgeon's practice here, and there's several good reasons why they would, as far as the challenge with the smoke and the workflow and so forth. What do you think is the number one reason physicians would move to this device over the others?
The main reason that surgeons change to any device is because they're safer and more precise. And that's the case with our technology as well. And we're actually not, the main change that we're making in their workflow is with our technology, they will not have to evacuate smoke. So with our technology, they don't have to worry about the excess temperatures, the smoke's not developed, so they don't have to wait for the smoke to clear. They don't have to use smoke evacuation systems. So it's just and our device functions just like their devices do. It just doesn't have all the bad side effects.
Using a different energy modality, do you think there's any risk of FDA requiring you to move to a de novo or PMA pathway?
No, there's no risk in that at all. The FDA actually loves us, partly because I worked with the FDA for about 15 years when I was at the University of Washington.
Okay.
But they recognize that there's a lot of problems in energy technologies and surgical devices. So the reason that we won't have to go to a de novo device is because there's a direct predicate, and that's the harmonic ACE. Even though the energy delivery mode is different, both our technology and the harmonic ACE use ultrasound transducers. So there are standards that we can compare to. And
Okay.
the harmonic ACE is the device that the FDA recommends we use.
Well, great. Well, you have advantages over, the current devices from Ethicon and Medtronic and so forth. But what prevents large incumbents from, replicating those same capabilities?
Well, we do have a very extensive patent portfolio. We have a nice moat built around our technology. But one of the main things is one of the previous presenters mentioned that, the large companies out there don't do innovation. They make minor modifications on the current technologies they have. They rely on companies like Novuson to do the innovation. So we've already had discussions with Ethicon, Medtronic, Ascalap, Hologic, Olympus. They're all interested in our technology. What they're waiting for us to do is get our first regulatory clearance and a little bit of clinical traction, and then they'll come in and acquire us.
Yeah, great. And what's your biggest challenge you face today?
Uh, the biggest challenge that we've always faced is just keeping a consistent flow of capital coming into the company to keep going forward.
Hmm.
Everything is pretty straightforward. There's very clear guidance on what you have to do with these devices to get regulatory clearance. Had a couple bumps in the road during the pandemic and stuff, but we're back at it and we're going forward.
All right, great. Well, thanks so much. That's our time. Appreciate a great presentation and good questions.
Thank you much.
With that, let's move to Pharma and Silica. Francois, if you want to launch your slides and kick off when ready.
Yes, sir. Now, Hal, I understand Kat is not there, so you'll have to be my lucky charm today.
I see.
How's that?
It looks good. Yeah, you ready to go?
No, I'm not. I don't
Oh,
know
I,
why.
I I got to the one screen there and didn't seem like it was ready.
Uh, Still working on it.
No problem.
How about that?
You don't see it now. We see you.
Not good for you.
Well, that was always my presentation to first time presenters. The slides are not the presentation. You are the presentation. The slides are supporting you. So I think you
Yeah.
had it there. We see it now.
Now, do you see full screen something?
Yes, we do. Chemo drugs.
Okay. Sorry about that, everyone. So, good afternoon. I'm François Arcan from Pharma in Silica. I'm here to present our precision chemotherapy. Its purpose is to radically improve the health and well-being of cancer patients and to create $1 billion of value in cost avoidance from the standard of care. I want to start by saying that we're at the IND enabling stage. We've visited the FDA already. We know our path. We're based in Quebec City, which is in the middle of a snowstorm right now. We have 100 interested investors that we identified with HAL's network and 4 Kiritsu chapters. We have a lead investor, term sheet, data room, and the due diligence is in preparation. Due diligence report. Sorry. Chemo drugs sell for $8 billion, and this is growing. You can't avoid it. It's used in combination with other treatments. It's a neglected market. Drugs are old and they're toxic. Just the paclitaxel, the biggest sales of cytotoxic drugs for 7 billion. They're dated 92 and 2005. It's an harsh artillery used by clinicians to carpet bomb the patient, hoping to reduce the growth of the tumor. It causes all kind of adverse effects that you have to pay as collateral costs to treat them, including hospitalization and $4 billion of sales for 2 drugs that treat anemia and infections. Our proposal is to avoid the adverse effects, saving these costs, thanks to UpPacli, it's an optimized paclitaxel. Imagine a very small truck that travels the blood without letting go of the drug and that releases it only when it reaches the solid tumor. It will bring cost savings for payers and for patients, superior efficacy, and well-being gains. We tested UpPacli in mice, and we've shown that if you do a 3-week, 3-injection regimen, with the same quantity of paclitaxel, the Taxol formulation, which is one of the standard of cares, will be exposed to the tumor for a lot less than 6 hours, while our UpPacli will be in the blood and exposed to the tumor during the full 21 days, which is a good predictor of higher efficacy because the drug reaches the tumor. But you might ask, if you have that big an exposure, could it mean more damage to the patient? Well, no. If you look at the purple bars, you see that the leukocyte, neutrophils, and lymphocytes are maintained, while with Taxol in gray, they're seriously depleted. As well, we've shown that on day 21, sorry, there's 5 times more paclitaxel in the tumor than Taxol. So what you have here is a de-risk platform for most of the chemo drugs, all of those that are hydrophobic small molecules. And we get this high level of blood cells, including red cells. And despite 21 days of circulation, you have safety. And the quantity of drug in the tumor suggests that we'll have also efficacy, which will be demonstrated in the coming months for the first tumor reduction study we're conducting. We're going to make money to our investors thanks to a two-step process. We're going to raise in the next year up to $10 million to complete a phase 1b/2a for solid tumor, one of the most toxic, lethal of the lung tumors, and preclinical proof of concept for 3 additional prototypes. From then, we're gonna license and propose manufacturing deals to Big Pharma, which should lead us in 2029 with exit opportunities at a value superior of $2 billion. The first thing we're gonna propose are the low-hanging fruits, which is the current program of Pacli and other cytotoxic drugs for solid tumors such as lung, breast, head and neck, and others. This will be offered to the companies that were making $1 billion 15 years ago selling these drugs, and those who currently sell the generic version for $100 million. The second lot will be for the cutting-edge oncopharma. We're going to propose to them to use the platform for their own novel candidates or failed programs, and we will also offer them our own prototypes, including super ADC or a nanoparticle that can carry 2,000 times more drug than an ADC, and multiple drug delivery strategies, and so on. The median value of those deals in phase 1 over the last 2 years has been $500 million in an all milestone perfect situation, but the upfront payment were between $28 and $58 million. This is where will be the beginning of the huge value gains to the investors, just the upfront payment. If you look at the progression of current treatments, you'll see that a superior safety and a inferior efficacy should position our Pacli in the hands of our pharma partners for a pricing close to what Abraxane got as a proprietary product. And if we are showing safety and efficacy in a superior fashion, price gained by the pharma partners should be in the area of Keytruda, which is about 20 times more than what Taxol gets and at twice what Abraxane gets. We raised $3.5 million so far. We've built our own production capacity, so we are autonomous in terms of preclinical and clinical lots. We also got an accelerated pathway from the FDA for our first product, and we are at less than 15 months to IND for non-small cell lung carcinoma. We also put together our team with the core values and core expertise in early stage drug development, nanoparticles, silica chemistry, and deal making. We have a lot of experts to complete these skills, and we're about to hire our own medical director and fractional experts, mainly from the US, in business development, regulatory, and pharmacoeconomics. We also filed 3 families of patents in 8 territories, of which 15 are already granted. 2 new applications are underway, and we protect seriously our industrial secrets, which is why we want to keep manufacturing in-house as long as possible. For a short time, investors can enter at the price of 2024, which is a 7.7 million valuation. Up to 1.5 million can be accepted at this price. The shareholders are accredited investors, plus and a 20% ownership by Quebec Economic Development Bank. There's no controlling shareholders. We manage everything by qualified majorities. In conclusion, our precision chemotherapy shows murine predictors of superior efficacy and safety. We have a fast track, which is saving us a year and $2 million in preclinical, issued IP, and milestones that are nicely placed over the next 3 years, which could bring successive value gains to the investors. We're going to hold a deep dive session with all our board members and shareholders and at least 100 invited investors on March 24th at 12 o'clock Eastern Time. And an invitation will be sent to anyone interested. Hal?
Great presentation. My question has to do around the nanoparticle safety. I mean, you're built on silicon nanomaterials, and they've had challenges in the past with accumulation, clearance, and so forth. Have you done any preclinical trials or tests to know that this is not going to be an issue for your application?
Well, we've shown that in rats, there's an excretion via the gastrointestinal tract. So the drug doesn't get stuck in the fingers and thumbs like other nanoparticles. We found that the particle doesn't collapse like liposomes and some polymer particles. I've shown earlier data that the immune system and red blood cells are not affected. So far, we've seen no traces of toxicity, no cytokine reactions. We've cut the organs and found no lesions or inflammation. So these are murine models, but it's the best you can get, and there's no issue of toxicity, despite the fact it's carrying poison.
Okay. Have you thought about turning this into a platform and applying it to other cytotoxic drugs later?
Actually, any small molecule that is hydrophobic is susceptible to be easily sequestered by the technology with minimal adaptation. We also believe that small molecules like kinase inhibitor will be relatively easily entered in the platform. If you want us to go into non-solid tumors, this will be more complicated. But just with the solid tumors we're looking after, you're looking at lung, breast, head and neck, maybe pancreas. So we have multiple billion-dollar markets in multiple territories for multiple potential drugs. So that's a lot and enough to get rich, I guess.
Okay. And that's good answer to the question. My final one is about manufacturing challenges. When you're starting to produce with silicon nanocarriers at GMP scale, do you see any challenges with the system you set up there?
Well, there are two aspects in your questions. First, you said GMP, which is a matter of control, and the second one is scale, moving from very small lab quantities. So for GMP, we went through all the hoops and loops of the FDA, and we're ready to present them our solutions, including final purification and sterility. So this is not a big concern, actually. As for the scale, the basis of the system is on 2 machines, one for production of the nanoparticle by sonication. The second one is by tangential filtration. And with the products we have, the tools we have right now, the same supplier is already producing larger scale machines. So we don't think that it's going to be easy because it's always big work to scale up. But we're not in a situation where we have to invent machines and processes. We've already increased by a factor of 40 our production capacity to deal with murine and rat studies.
Okay, great. Great answers to the questions. That's our time for now, but great presentation and appreciate that. I'm always learning more every time I hear the presentation. So I'm really starting to feel like I understand it. So that's great.
Well, Hal, if you want, you can invest.
We can talk about that. So next is BMI. Paul, if you'd like to launch your slides and kick off when ready.
Hi, Hall. Thanks for having us. Appreciate the time this evening. I'm going to walk you through how BMI Organ Bank is transforming transplantation, specifically within the kidney organ space. First, let me just give everybody a brief overview of the problem. There is a 10-year waiting list for kidney transplantation, and many folks on the kidney waiting list never receive a kidney transplant. The survival rates are fairly bleak if you remain on dialysis. However, if you are fortunate enough to get a kidney, your survival rate increases to 78%. The problem, though, is that there's plenty of donated kidneys. 90% of potentially donated kidneys never actually get transplanted. And the reason why they're never getting transplanted is because surgeons, as well as organ procurement organizations, don't have much information about the health and vitality of the donated kidney. And the reason why they don't have the health of this kidney is because they're using a technology that was approved by the FDA in 2003, something called the LifePort Kidney Transporter. And essentially what this technology does is it puts the kidney into a state of hibernation. It essentially is slowing the death of the kidney. There are no real-time metrics available for the kidney when it is on the LifePort machine. And so what surgeons and OPOs are looking for is they are looking for an ability to evaluate the organ function of the kidney, and they're also looking to for longer preservation times of the kidney. That is where the BMI solution comes into solving this problem. It is a closed loop system. We have developed in collaboration with Duke University a device as well as a solution that gives the kidney the nutrients that it needs. It's called the Duvall solution. It is a substitute for blood. We then provide the kidney with oxygen that keeps the kidney functioning, as well as we provide the kidney with a pulsatile flow or heartbeat, if you will. So there's 4 things that keep the kidney viable and alive, as opposed to standard of care: the Duvall solution, oxygen, and a pulsatile heartbeat. When you provide those things and are essentially mimicking Mother Nature, you can begin to evaluate the kidney with real-time vitals. There is a picture of our device on the left, and our technology allows for the kidney to produce urine. Once the kidney is producing urine, you can begin to run creatinine clearance tests. This is the same type of test that your physician will run when you have a physical. All of this has culminated to very good preclinical data relative to LifePort standard of care. Let me define the variable here. So delayed graft function is the probability that the patient post-transplant will go back on to dialysis. Standard of care today has the patient going back on dialysis 40% of the time. In our preclinical data, we have shown 0 evidence of delayed graft function. We've done this over 3 or 4 studies now. We just completed the GLP study that the FDA has asked us to do with the same headline results. I'll also note that this is the data that we presented to the FDA that gave us breakthrough device designation. Here is a pictorial of what we would consider to be a marginal kidney. These on the left is a kidney that a surgeon would often discard or not use because of the visual, subjective views that he sees of the kidney. Putting the kidney on our device, it clears up, as you can see in the middle. And then if you simulate a transplant, what the surgeons would say, I would have neglected to use the kidney on the left, but I would have transplanted the kidney on the right. We are in the process of preparing for our clinical trial. As I mentioned before, we've received breakthrough device designation in the third quarter of last year. We completed our GLP replication study in the fourth quarter of this year. We have some final FDA testing to do through the first half of 2026. We will file for our IDE in the third quarter of 2026 and expect our first patient to be transplanted late fourth quarter. Our business model is one of razor blade. We will be leasing the device to the organ procurement organizations as long as they guarantee a minimum utilization throughout the month. Each transplant will have a disposable and the Duvall solution. We'll charge $9,500 for that. It's a very simple market footprint, if you will. There's only 55 OPOs that we have to interact with. And this space is 100% reimbursed by Medicare. So day one of approval, we will have reimbursement coverage from the OPOs. Great preclinical data, multiple patents, large market, a $3.6 billion market, it's 100% reimbursable. There's been some interesting acquisitions in the space. We do have a pipeline. Technology can be used in liver, intestine, doing some interesting work in whole eye transplant, and we're working with the DoD on some limb transplants. Founders capital that's gone into the story, $3.7 million, grant funding, 8.9. We've raised 3.8 million of a 6 million convertible note. Total capital raised, 16.4 million. We are completing the seed round. We need another additional 2.2 of the 6. The convertible note has a 12 million valuation cap, 20% discount. National Kidney Foundation, Band of Angels, Golden Seeds, Mid-Atlantic BioAngels, Park City Angels all have invested in this current convertible note. The majority of them have due diligence memos that we can provide. Use of funds, the clinical readiness, the verification testing that I mentioned, the preparation for the IDE, and then working capital. We will have to do a Series A round after the close of this seed. We do have 2 lead investors identified for the $35 million Series A round, which will fund the clinical trial. Happy to talk more about those in further due diligence. Let me pause there, Hall, and see what questions you have for me at this time.
Yeah, great presentation. Francisco, Ning, if you have any questions, you're welcome to put them in the chat box. My question has to do around with the reusable base device and single-use consumable. It lists for $9,500 in revenue per transplant. And what's the manufacturing cost of those and the margins for that disposable unit?
Yeah, good question, Hall. We can manufacture the disposable unit as well as the solution for $3,500.
And what about scalability? What's the capacity that you have now, at which point you'd have to switch to something else?
Well, in terms of scalability, I'm not really sure I understand the question, Hall. We have a GMP certified facility that can produce enough capacity on the solution side for at least 35% of the market. As well as on the disposable side, we have a GMP manufactured that's got manufacturing capacity as well for about 30-35% of the market. So in terms of, you say we have to switch, I'm not really sure I understand the question.
Well, sometimes the manufacturing, the demand outstrips the capacity of the manufacturing facility because sometimes we're starting at a very small scale, and at some point it works up to like 1,000 units a month, and then we just can't produce anymore. And maybe the demand requires us to move to a larger facility. So now
We've
we
got
often have to raise money for that.
we've got plenty of capacity without the need to raise additional capital. If you think about 10% of the market, 10% of the market is about 4,000 procedures. So we can go up to about 35% of the market before we'd have to change manufacturing capacity. 10% of the market, 4,000 procedures, it's about a $40 million revenue business. Just to put that in perspective, Organox last year was acquired by Terumo for $1.5 billion. They do what we do in the liver space. They had about 75 million in revenue.
Okay. Is there any data play here? Are you capturing any data off the kidney as to its viability, the length of viability, and any metrics that could be reused later?
Yeah. So right now, current standard of care has very little data on the kidney. They capture flow, pressure, and resistance. We will be able to capture significantly more kidney vitals. We showed the urine output, and that's the first necessity, if you will, where you can begin to run vitals on the kidney. So it's a two-step process, Paul. And thanks for giving me the opportunity to walk this through. We'll be the first machine on the market that can capture additional assessment metrics. The clinical trial will then help us determine which of those assessment metrics give an indication for a healthy, viable, transplantable kidney. So first, you got to be able to capture the metrics, and then you can do the analysis on determining which of those metrics are relevant. And that is the step process we're in today, and we're the only company that has the ability to do that.
Are you planning to do anything with that data, putting it into an AI LLM of some kind or something
All of
that gives
that,
you
yeah.
the, yeah.
Yeah, all of that data will be stored in the cloud, and then ultimately analytics and statistics and the large language models will evaluate that data to determine which of those statistics, if you will, are relevant to provide the surgeon with a better predictive outcome of how that kidney will perform post-transplantation.
Do you see yourself monetizing that data, or is that just giving you a competitive advantage because you have that and others don't?
Just a competitive advantage relative to what others don't have.
Okay.
And remember, the current standard of care can't even capture that data. They're putting the kidney in a state of cold hibernation, and when the kidney is non-functioning, it can't produce the data. You can't collect any statistics on that kidney.
So you're expanding the use of kidneys now through this process. Any idea of what the expansion rate is that you're predicting with the use if it's fully deployed?
Yeah, so currently in today's market, there's 21,000 kidneys that are transplanted from deceased donors. There's about 6,000 to 8,000 living donors that donate their kidneys. But if we're talking about deceased donors, there's about 21,000 to 22,000 kidneys transplanted every year. The OPOs actually procure about 31,000 kidneys, and about 9,000 to 10,000 of them are actually discarded every year. And the reason why they're discarded is because the surgeons don't have enough information about the kidney to make an informed decision on whether that kidney will be viable for transplantation. And so the first thing that we will provide the surgeon is, and the OPOs, is those 9,000 kidneys that they're discarding every year. We should be able to give a window into whether those kidneys are actually viable. And there's some good data that says that about 60% of the 9,000 are actually viable kidneys every year. So we'll be able to tackle the 20, 21, 22,000 of actual kidney transplants already. We're going to grow the market by capturing part of the 9,000 that are discarded and not used with our technology.
Okay, great. That answers my questions. I want to thank you for a great presentation there, Paul, and I want to thank all of our presenters for doing well today. We will take the recording and the slides and send it to the investors and capture interest and feedback to bring back to you. With that, we're at the end of our time. Thank you guys so much for joining today. Thank you guys for online as well. And with that, we'll go and close it out and looking forward to seeing you guys next time. Hope you guys have a good day. Bye-bye.